Today I have a really cool bit of science news for you. Researchers in the Department of Biochemistry and Microbiology at Rutgers, New Brunswick, wanted to look closer at a species of sacaglossan sea slug. These sacaglossan sea slugs are known for their algal plastid symbioses, wherein the sea slug can absorb plastids from algae and somehow integrate that into its tissues or into its metabolism. The particular sea slug of interest is the species Elysia chlorotica, which lives along the coasts of New England, eastern Canada, and Florida. In appearance, it looks like a large, thick piece of lettuce, and on the end of the stem, or on the end of the main vein, is a weird gastropod face. The E. chlorotica is a vibrant green because its cells are packed with these chloroplast plasmids that it steals from the algae. It feeds on a type of algae that grows in clumps of long strands, and the cells in these strands don't have hard walls dividing them, kind of like a cenocytic fungus. So the slug is able to grab a bundle of these algae strands, bite off the ends of them, and then suck out all of the stuff inside, including the plasmids. During digestion, the chloroplasts are identified, and they're protected. Instead of being broken down and digested for nutrients like everything else, like the algal cell nucleus, the cell membranes, and the proteins in the cytoplasm, the chloroplasts get removed from the digestive system, and they get placed in a storage organ lining the intestinal tract. From here, they permeate into the rest of the animal's body, turning its cells green and making its tissue photosynthetic. Professor Debashish Bhattacharya was the senior author of the study on these sea slugs, which was published in the journal Molecular Biology and Evolution. When Bhattacharya is talking about the Elysia chlorotica sea slugs, he says, and I quote, "It has this remarkable ability to steal these algal plastids, stop feeding, and survive off the photosynthesis from the algae for the next 6 to 8 months," unquote. To perform photosynthesis with stolen chloroplasts for that long, it's kind of weird. It's mysterious, because the algal nucleus was destroyed during digestion. So the question is, where exactly are all of the proteins coming from that would maintain these chloroplasts, that would keep them healthy and regulate them and keep them functioning properly? And for that matter, where are the proteins coming from that take the chloroplast's photosynthetic byproducts and turn them into sugars? How is all of this happening? Where are these proteins coming from? Where are the genes for these proteins embedded? To examine this mystery, the researchers sampled cells from the sea slugs, and they used RNA sequencing to see what kind of RNA was getting expressed, like the messenger RNA that links DNA to protein. They found, and I quote, that upon exposure to and ingestion of V. litoria plastids, genes involved in microbe-associated molecular patterns and oxidative stress response mechanisms are significantly upregulated, unquote. Now, this kind of makes sense. Once they come into contact with these plastids, their molecular pattern recognizing systems would come online. And also, it's my understanding of the data that the sea slugs themselves seem to have a few genes that can participate in the photosynthetic process. But when they say oxidative stress response mechanisms, that makes me think that their participation is limited to just a kind of damage control to minimize unintended oxidation. As Bhattacharya explains, these findings help shine more light on what's going on, but we still don't understand how the chloroplast can be maintained for so long. That mystery remains, but Bhattacharya wants to figure it out, because the implications of that knowledge with regards to technology and human civilization are, in his opinion, and I tend to agree with him, tremendous. He says, and I quote, "The broader implication is in the field of artificial photosynthesis. That is, if we can figure out how the slug maintains stolen, isolated plastids to fix carbon without the plant nucleus, then maybe we can also harness isolated plastids for eternity as green machines to create bioproducts or energy. The existing paradigm is that to make green energy, We need the plant or algae to run the photosynthetic organelle. But the slug shows us that this does not have to be the case." Unquote.